and good afternoon. I will go briefly through the suggested topics in the template of the presentation. First, what we are doing, what we are offering. Our main business is next-gen sequencing, which is the current main genomics method. And in this field, we offer services both just sequencing on our sequencers, which are currently Illumina NextSeq and Illumina MySeq machines, the smaller ones. And we also offer library preparation services for either standard approaches like capture-based enrichment, DNA library preparation, RNA sequencing, different types. Now starting with single-cell sequencing for transcriptomic experiments and others. It depends on customer needs. Our second target is flow cytometry. We offer access to flow cytometer and also offer a service of cell sorting. And we have also available some fluorescent microscopes, either with microdissection or with continuous imaging. But microscopy is not our main topic. These are our main services, which are provided as a service, and mainly for internal Cytec users, we offer access to our instrumentation, which is mostly real-time PCR machines, but also flow cytometer or instrumentation for sample quality control, like fragment analyzer, tape station, automation. We have pipetting robots, both pre-PCR, post-PCR, and also some microscopes. Another suggested topic was new instrumentation, because most of our instruments were bought before 2015. Now we depend mostly on research infrastructure projects, which we are connected to 2 of them. And from this, we've been able, unfortunately, to buy only one quite small instrument, which is 10x Chromium. 10x Chromium. This is microfluidic controller for preparation of single-cell libraries in droplets, which is current state-of-the-art until next year. It's the state-of-the-art. Next year there will be new, better, bigger machine for single cells. What our plans is to get somehow Illumina NovaSeq. NovaSeq is the largest sequencer from Illumina portfolio, and what is important on why we want it is because it dramatically decreases the sequencing cost. It depends on case, but compared to our NextSeq system, the price reduction could be from 40 to 90% per base or per read, which is really a dramatic one, and we can get several times more sequencing and more data for the same price with this instrumentation. But unfortunately, it's not possible to do it from research infrastructure budget in this investment window, which was not optimal for us. So we are trying to get financing from it somewhere else. So hopefully it may happen in the middle of next year. Another nice, but in this case much smaller planned investment is this small long-read sequencer, single molecule sequencer from Meteon, from Oxford Nanopore, which is a nice small machine with capacity up to 100 gigabases of data from per flow cell. This machine was announced this summer and could be available on the market during next year. And we are thinking about it because it's quite cheap. It's only 50,000 euros. So it may be possible to invest into this machine, which I have to say that this technique is used by many of our users, now mainly with MinION sequencers, cheap machines that are, and compared to these cheap machines, it offers again much better price per gigabase than MinION. And as I said, we are dependent with investments mainly on research infrastructure related projects. We have spent all the money for current investment window, so we are waiting how it will look in the period 2023 to 29. Question about data policy. We decided to share the data policy with Core Facility Bioinformatics because logically the data flow is from our sequencers to Core Facility Bioinformatics, which handles the data analysis. Briefly, we have different data policy for different type of data. For data generated on small instruments like qPCRs or sample QC control machines, it's user responsibility to take the data and save them themselves. Different is for our main next-gen sequencing data, which are stored centrally at backed and archived storage and are there for at least 6 months. Current real politics is until we have space, and we still have space for, let's say, next 2 years. So we expect the life of the data could be, availability on this storage could be up to 5 years, but we guarantee 6 months. Results of user survey. I just selected just text notes that were there because it took my attention. First is, then it appeared there at least 2 times, questions on single-cell RNA sequencing and spatial transcriptomics. As I said, we bought the 10x Chromium controller, which is state-of-the-art machine. It's really easy to handle the machine itself and prepare the libraries. Now we are running experiments for our customers in at least 3 different projects. We started with some easier things, mainly blood cells, but now also starting with organoids and other types of cells. So we are learning how to prepare the sample correctly because it's the main problem. Prepare the sample to be viable, to be really single cell suspension, and to do it cost effectively. Because single 10x library preparation is around 2,000 euros just in the chemistry. So we don't want to waste too much money. And second question was spatial transcriptomics, which is now really popular. Unfortunately, there is now only, there are probably different vendors, but one of them is the main one. So there is a monopoly, and that's why this approach is quite expensive. So we are now planning and preparing our internal pilot project to get some experience with this technology, because it's, again, very difficult to prepare the samples properly. And even I wrote there, super expensive compared to single cell. Single cells are cheap compared to spatial transcriptomics. So the single experiment is like 8,000 euros in chemistry. It's up to 4 very small samples. So that's why we want to test it on our samples first, to invest. Another sentence I saw there is closely connected with this reinvestment politics. They should buy at least one more flow cytometer machine because it's really heavily used. I have to answer that we would like to, but we are limited with this available money. Or we can, flow cytometer is not so expensive machine, not so expensive, it's several million crowns, but still available from project money and other sources. But it would mean that we would require our users willing to use the cytometer to really pay for it and help us with covering of the expenses, which we see, at least from personal discussions, some resistance in paying money for instrument access. At least the money that are needed for buying the machine from internal sources. And question for trainings. We offer basic trainings, how to use the labs and how to use the main self-used instrumentation, which are qPCRs. So we train people how to use the qPCRs the way not to break them, not to destroy them. And these are requests what the users would like to see. We like to have trainings, but especially for these more difficult parts, we are now thinking how to do it to be accessible to people, not to, because it costs quite a lot of money to run the experiment, and with running it in theory is not training. Training is hands-on training. You should pipette, you should test the system and see the output. So currently we would like to cooperate with some vendors, especially with Genetica and Tenix for single cells, or prepare some really just user-paid training for library preparation. Acknowledgement, you've heard it in all the presentations. I just have to ask also to help us get money from Ministry of Education and acknowledge support of our core facility. We are connected to 2 infrastructures, which is not usual. So for NGS-based services, we ask to acknowledge National Center of Medical Genomics, and for flow cytometry services, Atris, CZ. Most important results. Be honest, I'm really happy that we have still people now understand why we want acknowledgements, and there's now it's the number of acknowledgements for our core facility is still increasing, slowly but increasing, and I'm happy that even internal users started to acknowledge our core facility. I haven't selected any paper because it's mainly our users' paper, and I think most they think they are important, and I don't want to select any of them preferentially. What I'm also happy is we are still able to grow a bit. This is expressed in money spent by our users. This is just for the sequencing chemistry. So you see that even these are data until the end of August. So until the end of August, we spent more than last year and the previous years. And successful implementation of single cell methodology. It's for our internal purpose also important. Thank you for your attention. Questions?
Yes,
We are using external machines in collaborating institutions. So it's possible, but it's the comfort for us and for our users, it's not, the planning is a bit more complicated.
Okay, I will go back again to plants. Have you ever tried that flow cytometer for plant cells? Do you work with the plant cells?
Tomáš?
Yes.
And did you try or just?
Yeah, tried.
Okay. So, yeah, so you have some devices to use it for. Okay.
I think, I don't know, it was, you can prepare the single cell suspension, it's called plasmoids or something from cells.
Protoplasts.
Protoplasts. But probably not. If you can prepare this, then probably should be no problem.
Yeah.
Hi, one more.
Nanopore technologies recently announced this fungal sequence output of data.
If you are interested, you can get in contact with my colleague. The fungal is quite cheap, but the throughput is quite low, and what we have found, just very short shelf life. So all the 12 fungus should be used within 3 weeks of delivery.
Really?
Yes. Unfortunately, we found it after ordering and running some of them. So
It seems great, but hands-on experience.
And again, these machines are maturing, but again, the library preparation is the key. So now we have users that were able to get 22 gigabases from Minion, which is really nice, at least for the price. And with this fungos, it's cheap, but you have to buy at least 12. So you have to spend at least $1,200, I think, or euros. I'm not sure. And one fungo is $100, and the shelf life is not so good. But if you plan some optimizations, and so it may help. But this Promethean P2 may be better. You can expect that the flow cells will be quite expensive because you will not buy them in volume, large volume. But the machine itself, it's not so expensive. And now we also have available direct online base coding for Minion on graphic card. So now you can do all these stuff that require reading of the sequence during the sequencing. So it really works nice. So if you have experience with Nanopore, and base coding can take up to a week on your computer. Now it's running really in real time together with the sequencing, and it's working quite well. It depends. If you do consensus, then it's 0.1%. But you have to do consensus and sequence in depth. Otherwise, what I see these indel type errors are there in every read, several of them. So 5%, I would guess. So as I said, this user that had these long high output was also able to produce. Then they had the N50 around 12 kilobases, which is fine for helping with the genome assembly, and 22 gigabases for $1,000, which is really nice, I think. But it was in the library prep, it was not in the sequencing. So they did the library prep very well.